Okay, let's watch this little video. It's nice to have an animation that kind of summarizes everything we just finished talking about.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
All right. So the spindle, what we call the mitotic spindle, is made out of microtubules. Some of those microtubules are attached to the chromosomes at the centromere, and some of those microtubules are just attached to other microtubules and are going to be used for lengthening that spindle. The centrosome is the microtubule organizing center. In animal cells, primitive plants like ferns, primitive fungi, and many protists, they do have centrosomes. This is what one centrosome is made of two centrioles. They're at 90 degrees to each other. They're made of microtubules, and they are at the center of the aster in the mitotic spindle for cells that have them. For most plants and fungi that don't have centrosomes, they still have a microtubule organizing center. It just doesn't have centrosomes at the center. The last thing that happens in interphase before mitosis starts is the replication of these centrosomes. So two things happen during interphase. Replication of the DNA happens during S phase of interphase. Remember, interphase has G1, S, G2. Those are the three phases of interphase. So G1 is normal metabolic activity of the cell. S is the synthesis and replication of the DNA. S is for synthesis. And G2 is also normal metabolic activity and gene expression. But if the cell is going to divide and it has a centrosome, those centrosomes are going to replicate before we go into mitosis. So those microtubules are going to attach to a special protein complex that is bound to the centromere of the chromosome, and we call that the kinetochore. Kinetochore is a large protein complex. It's attracted to bind to the specific sequence and proteins of the centromere. So remember, the chromatid, that long piece of DNA that's wound around proteins, that's our chromatid. And the centromere is a specific repetitive region of DNA. There are no genes in the centromere. And it's a particular repetitive sequence, and that sequence is what gives it the ability to attract particular proteins to bind and the kinetochore proteins to bind. So if the DNA of the centromere becomes mutated, if it's mutated enough, and mutate again means change to the DNA sequence. So if that sequence changes enough that kinetochore proteins won't bind there, that chromosome is going to be lost during the next cell cycle. Okay, so microtubules bind to the kinetochores, and at metaphase, the chromosomes are being pushed and pulled by those microtubules, and they will end up lined up along the metaphase plate. So there's two identical sister chromatids. They each have a kinetochore and a microtubule attached to them. So here's a little video. I'm going to have to go back to get rid of my laser pointer so I can click on it. This is a time-lapse video of an animal cell. I don't know what kind of cell it is. The source that I got it from didn't say. It's not a human cell because there aren't nearly enough chromosomes. It's either a fish cell, it could be a pufferfish cell, and it could be a Drosophila cell. I can't tell from the context here. There's no indication of the size of the cell, which would give me a clue as well. But here we can see the outside boundary of the cell and the nucleus on the, oh wait, let me go back to my pointer here for a second. Okay, so here's the nuclear membrane here, and then the larger cell is about yay big there. And what we're going to see is the nuclear membrane is going to break down. You'll see the chromosomes condense, and then they will separate, and then two new nuclei are going to form. Lacey, shh, shh, that's enough. And two new nuclei are going to form. Nothing is stained in here, so we won't be able to see the spindle. There's no fluorescent staining. This just uses light. This is a high contrast microscopy image that starts with just natural light. So that's why it has that kind of three-dimensional effect. All right. So here we go. We can see that it's prophase, early prophase. Now it's late prophase. We can see that nuclear membrane disappear, and the chromosomes are condensing and getting shorter. Now they're being pushed and pulled by the microtubules. They're jiggling around. They're going to eventually end up at the metaphase plate. We can't see the microtubules in this preparation. Still prophase. Still prophase. Metaphase. Anaphase. Still anaphase. Telophase. And cytokinesis. All right, let's watch that one more time and pay attention to how long these stages are. So the time lapse is steady here, so that means it's relatively, we can compare the times of the different stages. So prophase is really long. 80 to 90% of the total time the cell spends in mitosis is spent in prophase and prometaphase. All right, so we're already in prophase here. The chromosomes are already condensing, so we've missed part of prophase already. Prometaphase or late prophase, still prophase. This entire process would take at least 90 minutes in an animal cell, so this is very sped up. Still prophase, still prophase, metaphase, anaphase, telophase, cytokinesis. So our mitotic spindle here, so we have two kinds of microtubules. Oh, wait, I need my laser pointer back. We have two kinds of microtubules. We have microtubules that are attached to the chromosomes at the kinetochore, and here's a nice TEM microscope image of that with our, you can see that there's many microtubules attached to this one chromosome here. And then there are other microtubules that are not attached, and in fact they are just attached to other microtubules. And these microtubules are how that spindle extends. If you remember that Drosophila video that I showed you where the microtubules were labeled with a fluorescent tag, during anaphase and telophase, that spindle is stretching out and getting longer, and that's due to these microtubules walking away from each other using the same motor proteins that are moving the chromosomes along the microtubules as well. So how do we know which end of the microtubule is shortening? Because there's actually two possibilities here. One possibility is that the chromosomes are being reeled in like a fish on a line, and it's this end that's shortening. The other possibility is that the chromosomes are kind of chewing their way up the microtubule and it's shortening at the chromosome. So where is it shortening? Is it shortening here or is it shortening here? So a very clever experiment which revealed which of those was occurring. They used fluorescently tagged cells, so the microtubules were lit up, and then you can take a laser and ablate that fluorescence so that now you have portions of the microtubules that are dark. So with that ablated region, you can ask, okay, what happens to this mark during anaphase? If the chromosome, if the microtubule is shortening at the aster and being reeled in like a fish on a line, then the dark mark should move. If, however, the microtubule is shortening at the chromosome, the mark should stay in the same place and eventually be overtaken by the chromosome. And that's the second one is actually what is happening. So the microtubule is shortening at the chromosome. It's not being reeled in like a fish on a line. We often think of it as the chromosomes are being pulled to the poles. But actually what's happening is the chromosomes are chewing their way up the microtubules towards the poles, which is kind of cool. So we now know that what's actually happening in that kinetochore. There are two things happening in the kinetochore. One is the kinetochore contains dynein-like proteins that are walking on the microtubule. If you remember how vesicle transport occurs in the cytoplasm and how cilia and flagella move, we have proteins called dyneins that can walk on the microtubules, and that allows cilia and flagella to bend. It also allows vesicles to be transported along those microtubules using these little dynein feet that can walk on the microtubule with ATP as the energy source. Well, this does the exact same thing. Inside the kinetochore, we have these same little motor proteins that can take a step. They take one step per ATP. And at the same time, the microtubule itself is being disassembled. It's being taken apart. So as the chromosome is walking up the microtubule, the tubulin subunits themselves are being actively removed and disassembled. So it's kind of, the chromosome's kind of chewing its way up the microtubule. The non-kinetochore microtubules, the ones that are just overlapping and are not connected to a chromosome, those are walking apart from each other using those same dynein proteins. Cytokinesis is the process of the separation of the cytoplasm and the membranes. And it occurs differently in plants and animal cells. Animal cells don't have a cell wall. They can change in shape quite easily. So cytokinesis occurs by a different mechanism. So in animal cells, and this is a nice scanning electron microscope image here, it's a fairly large cell. I mean, look at the size here. This is 400 or 500 micrometers in diameter, so that's a huge cell. So this is definitely not a human cell. It's probably a fertilized egg of some kind, making its first cell division from that size. It could be it's a very small amphibian embryo, or most likely a fish embryo, judging by the starting shape as well. Okay, so the way an animal cell divides, when cytokinesis occurs, it uses actin, which is what microfilaments are made out of. I've been calling them actin filaments because that's less confusing. Microfilaments are easily confused with microtubule. So actin is, remember, under the plasma membrane in a net, kind of in the entire cell. Anytime the plasma membrane is moving, like an amoeba that's moving by with pseudopodia and crawling, that's using actin filaments. And the same thing here. So there's a kind of a purse string of actin filaments that contracts and cinches the cells apart. Now, plant cells don't have that option. Plant cells have a rigid cell wall, and they are constrained in shape change. They can't bow out as they are cinched apart. So instead, plant cells form a cell wall in between by vesicles fusing together. So vesicles accumulate at that cell boundary. This cell here is in telophase. The new nuclear membrane is forming, and so is the boundary between those two new cells. Cytokinesis is happening at the same time. So cytokinesis is vesicles. Each vesicle fuses together and forms one continuous membrane. And then the cell wall is secreted in between those layers. These are examples of images from a plant root tip, one of the things that we're going to look at in lab. And we can see that we like looking at plants first because plant chromosomes are much larger and fluffier and easier to see than animal chromosomes in general. So it's nice to start with those because it's really easy to identify the stages of mitosis. So here we have prophase. We can see the chromosomes starting to condense. I see. Shh, that's enough. Shh. We can see the chromosomes starting to condense, and the nuclear membrane is still intact. Late prophase or prometaphase, we see more condensation, and the nuclear membrane is breaking down. Now in plant cells, we cannot see the spindle very well. So that's why we also like to look at animal cells, because even though in animal cells the chromosomes are smaller, it's harder to identify the stages of mitosis, we can see the spindle because it's denser in animal cells. Okay, metaphase, all of the centromeres are lined up on the metaphase plate, but the chromosome arms are going in every direction. Anaphase, we can see these are separating, and then telophase, we can see the nuclear membrane is reforming. Now, how do I know this is telophase and not the next interphase? Because I can still see the spaghetti strands of those chromosomes. Once it's back in interphase, the nucleus is going to again look like diffuse sand. In telophase, we can still see the chromosomes, and in plant cells, it often looks just like this, where the chromosomes kind of look like fingers, like they're rolled up during telophase. And we can see this very faint new cell boundary starting to form here for cytokinesis in the plant cell. How did mitosis evolve? So prokaryotes, which were the only life forms for 2 billion years, and they're still around, so we can study how they divide. They have a very simple type of cell division that we don't call mitosis, we call it binary fission. And they have a single chromosome which replicates, and then the two chromosomes are pulled to different ends of the cell, and then the cell divides. So how did we go from this kind of simple partitioning of the cytoplasmic contents to a complex system of breaking down the nuclear membrane and creating the spindle and making sure that all, one copy of every chromosome ends up in each daughter cell? Well, one of the ways to look at that is to look at the equivalent genes in bacteria. So now that we have good DNA sequencing, cheap DNA sequencing, this is possible. So here we have the three main proteins that are involved with eukaryotes. Tubulin, that's the subunit, that's the protein that makes up microtubules. So there's gene for, there's actually two different ones, genes for tubulin proteins for the alpha and beta subunits. For actin, there's a gene for actin, that protein, and then intermediate filaments are made out of keratins, and we have several different genes for various keratins that make up the intermediate filaments. Remember that intermediate filaments are what we often call the boring part of the cytoskeleton because they're not involved with movement of any kind. Tubulin is involved with moving chromosomes, moving vesicles around, cilia and flagella. Actin is involved with cells that crawl and use pseudopodia, and also cytokinesis in animal cells. Intermediate filaments are most involved with just maintaining the cell shape. So if we search bacterial genomes and ask, okay, do they have equivalent genes to this? I mean, we didn't, before this, we didn't really think that prokaryotes had a cytoskeleton, but do they have the genes and therefore a protein that is related to the eukaryotic cytoskeletal proteins? And the answer is yes for all three. So they have a gene that is in sequence similar to tubulin, FUTZ, and in fact, we do find that this protein is found during cell division, during binary fission. They have an equivalent of the actin gene called MREB, and in fact, we do find that this protein is localized during binary fission as well. And they also have a protein, this CRES, that's the equivalent of keratin, and it is not involved with binary fission, just like intermediate filaments are not involved with mitosis, and it does seem to help with maintaining the cell's shape. So interesting story, and the fact that prokaryotes have the similar related proteins to the proteins that are involved with the cytoskeleton in eukaryotes. There are several different types of mitosis. We've been talking about this last type here, where the nuclear membrane completely breaks down, and then a spindle forms, and then the chromosomes separate, and then the nuclear membrane reforms. That's what most eukaryotes do. Plants, animals, most fungi, most protists have that type of mitosis. There are a few weird variations, though, and like diatoms, the nuclear membrane doesn't break down. Instead, the spindle forms inside the nucleus, and the same with dinoflagellates. Microtubules extend into the nucleus during mitosis. So is one of these the ancestral type of mitosis? And scientists think probably not. That probably the type that our cells use, the most common type, this is probably the ancestral type. And these are weird other types that evolved later. The cell cycle is regulated. Cells don't want to be dividing as rapidly as they can all the time. So bodies need to control when cells are going to divide. So eukaryotic cells require a signal in order to divide. And we'll talk a little bit about cancer cells, which you probably know. One of the hallmarks of cancer cells is that they divide when they shouldn't be dividing. So normally, animal cells require a growth factor to divide. If cells do not receive a growth factor, then they will not divide. And they have receptors on their surface for these growth factors. If they don't receive any growth factor, now what's a growth factor? A growth factor is a molecule that's secreted somewhere else. It could be a neighboring cell. It could be a cell in another part of the body, like thyroid gland. If they don't receive some kind of a growth factor, many of them are peptides, they will stay in G1. And remember, G1 is before DNA synthesis. So they won't even replicate their DNA without the right signal. And if they're going to stay in G1 permanently, then we say they're in G0. And cells that would never divide would be things that are what we call terminally differentiated, like muscle cells and neurons. They are not going to divide again because they're too differentiated. They couldn't go through mitosis. Growth factors are small proteins, which we call peptides. Things that have fewer than 100 amino acids would be considered a peptide. And many growth factors are small proteins. Things like insulin and human growth hormone, those are peptide hormones that behave like growth factors. You probably know insulin is more familiar to you as something that helps regulate blood glucose, but it also behaves as a growth factor and stimulates cells to divide. Most cells can only respond to 1 or 2 or 3 growth factors, depending on the cell type. So there are some general growth factors that many cells will respond to, like human growth hormone and insulin. But there are other growth factors that are very cell type specific, like only for neurons or only for epithelial layers. So there are tissue specific ones as well. So if we go back to our cell cycle here, so interphase is much longer than mitosis in eukaryotic cells. So in interphase, G1 is when the DNA has not even replicated yet. S, or for synthesis, DNA synthesis specifically, that's when the DNA replicates, and then G2. Now cells have to receive growth hormone during G1 or they will not go through S. And then they have to continue to receive hormone signaling in G2 or they will not go on into mitosis. So, and the way this was discovered was by attempts to grow human cells and other animal cells, notably chick cells, in culture. And if you try to grow cells in culture, if you take isolated tissue from humans or chickens or mice and you put them in a medium, this is a typical looking tissue culture container here, it's a flat bottle. And if you put growth media in there that contains glucose and the right salts and amino acids and everything else that cells might need to grow, they won't die, but they won't divide. And they will just sit there. And it was discovered that you had to add something to them that contained these small peptides. And initially that was blood plasma. If you add that, now you've added a growth factor, and now your cells will divide. PDGF was one of the first growth factors discovered. PDGF stands for, oh, there, now it just went out of my brain. Not plasma-derived growth factor, it's the little things that clot your blood. I'll think of it. But this was derived from blood plasma, and when it was added, it contained PDGF, and that allowed the cells to divide. It provided a growth factor for them to divide. But it takes more than that to control the cell cycle. So cells also will not divide if certain situations are present, even if they have growth factor, even if the growth factor is telling them to divide. And one of those external signals, we call it density-dependent inhibition. Platelet-derived growth factor, that's what it is. The P stands for platelet. Platelets are those little cell fragments in our blood that help blood to clot. So PDGF is platelet-derived growth factor. I knew it was in my brain somewhere. So remember, inhibition means stop. So density-dependent inhibition, this means that if cells are too crowded, that they will not divide, even if there's growth factor present, even if there's plenty of food, oxygen, everything else that the cells need. And animal tissues depend on this, that even if there's growth factor present, once the cells start to become crowded, they will stop dividing. Animal cells also exhibit, almost all animal cells, exhibit anchorage dependence. So anchorage specifically to the extracellular matrix. So we talked about the extracellular matrix, which is collagen is an important protein that's in the extracellular matrix. The extracellular matrix is what is surrounding animal cells in general, human cells as well. And cells need to hold on to a substrate in order to be happy and divide. Most cells. But things like cells in epithelial layers, if they don't have extracellular matrix to hold on to, they will also not divide. So if cells break away from a tissue type and they end up in the bloodstream or they end up in the lymphatic system, they're not going to divide because they don't have anchorage. They don't have their extracellular matrix. Cancer cells often lose both density-dependent inhibition. Cancerous tumors can be extremely dense and often feel hard and rock-like because they're so dense. And they often also lose anchorage dependence and can get into the bloodstream or the lymphatic system and move to other places in the body, and they will continue to divide while they do so. So for anchorage dependence, so if we have our cells in our tissue culture bottle and we don't provide them with some kind of an artificial substrate. So typically when you're doing tissue culture, the bottom of your culture dish would be coated with some kind of a protein like collagen or fibrinogen, something that is going to allow the cells to feel like they have an anchorage. And then they will divide. And they'll divide until they fill up the dish. And once they become dense, then they will stop dividing, even if you continue to provide growth factors. If you remove some of the cells, they would divide a few more times till they fill up the dish again. Cancer cells grown in culture do not follow either one of these strictures, and cancer cells will form a large dense mass, and cells in the center will even die from lack of nutrients and become necrotic, and the cells will continue to divide. So cancer cells don't normally respond, although many cancer cells still require growth factors. They get around the need for growth factors in a few ways. Sometimes cancer cells contain many mutations, as we've talked about before. That's the starting point for cancer, is mutating the DNA in the cancer cells, whether we're talking about the nuclear DNA or the DNA in the mitochondria. Cancer cells have a lot of mutations. Not just a single mutation, lots of mutations. And genes can get turned on that shouldn't be turned on in that particular cell type. So often cancer cells make their own growth factor, so they secrete the growth factor and respond to the growth factor at the same time. Sometimes their receptor for the growth factor, the little receptor that receives the signal, sometimes it's mutated so that it responds as if it always has growth factor present, even if there isn't any growth factor. And sometimes they just seem to operate outside of any need for control of their cell cycle. When they do respond to a growth factor that we can remove, like estrogen or testosterone, that's often a good thing, because we can remove those growth factors temporarily from the human body without causing significant harm to the person. Other growth factors we cannot remove because it would seriously harm the person that's using them. So the process of going from a normal cell to a cancerous cell that's going to divide out of control, this process is called transformation, and it's been extremely intensely studied, but is still not completely understood. Why you can have cells that have many mutations and are not cancerous, and other cells that have similar mutations that do become cancerous. We do know that the immune system is important for eliminating precancerous and cancerous cells, and anything that interferes with the immune system, taking immunosuppressive drugs, if a person has a disease that involves reduction of efficacy of the immune system, those individuals are at higher risk for cancer. For example, individuals who have had a kidney transplant and they have to be on immunosuppressive drugs, which lower the activity of their immune system. Those people are at higher risk for cancer. So that tells us that the immune system is normally important in eliminating cancer cells. How would it do that? Well, cells with mutations are going to have often abnormal proteins on the surface of their cells, and our immune system will recognize those abnormal proteins and attack the cells as if they are not self. And this is another reason probably why young people for cancer is so rare in young people. So when you're in your 20s, that's the lowest risk time of cancer in an individual's life, is when they're in their 20s. That doesn't mean that they'll never get cancer, but it's very rare for people in their 20s to get cancer. You're at a higher risk of cancer when you are elderly, because your immune system is less effective, and it slowly declines as you get older. Now, just because a cell divides too much doesn't mean that it's going to be cancer. There are a huge category called benign tumors. There are many kinds of overgrowths of cells that are not cancer because they don't invade neighboring tissue. So an example of a benign tumor would be a wart. A wart is an overgrowth of skin cells, but it is benign because it's not invasive. And you can in fact remove warts by cutting them off, or by burning them off, or freezing them off. And as long as you kill all the original cells, they haven't invaded the neighboring tissue, so it won't grow back. But we do know as well that cancer starts with damaging the DNA. Cancer cells have many, many mutations, and they continue to divide abnormally and accumulate more mutations. So in a cancer, if a person, by the time a person is diagnosed with a cancer, their cancer cells are a heterogeneous mix of cells with slightly different assortment of mutations. The cancer cells are not all identical because they continue to accumulate mutations abnormally. One of the things that was discovered early on was that normal cells, most normal cells, have a limited number of cell divisions. And the person who discovered this was a guy named Leonard Hayflick, and we call this limit in the number of cell divisions that will occur the Hayflick limit, after him. And what he discovered about human cells was that no matter how young the person was that you got the cells from, human cells, and there are human cells that don't obey the Hayflick limit, but most human cells will only divide a maximum of about 50 times, and then they will stop dividing. And in culture, you can get this to happen in about a year of dividing, if you get the cells to divide as rapidly as they will. And Hayflick did not know the reason for this limit, but we do now. Now we know it's due to telomeres. So the telomeres are the ends of the chromosome. We talked about the centromere. The centromere is a region of the chromosome where we find repetitive DNA that attracts particular proteins to bind, that will help kinetochores to attach to the chromosome so that they will divide during mitosis. The telomeres are at both ends of the chromosome, and only eukaryotic chromosomes have telomeres. Prokaryotic chromosomes are a circle, so there's no ends. Telomeres are also repetitive DNA, like the centromere, and it varies between species somewhat exactly what this repetitive sequence is. This is the human telomere sequence. And what happens is, so there's no genes in the telomeres, just like there's no genes in the centromere region of the DNA. So this end, which is these repeats are repeated thousands of times in the end of the telomere. Normally, when cells divide, the DNA polymerase, that's the enzyme that replicates the new strand of DNA, the two strands of DNA, the two strands of the double helix separate, and DNA polymerase makes a new copy of each one. So we get two new double helices. Each one has one new strand and one old strand. So every time that happens, the telomeres get shorter on one strand, on the new strand, because the DNA polymerase has to sit on the DNA in order to add nucleotides and make a copy. So that means when the DNA polymerase gets out to the end, it can't replicate the DNA that it's sitting on. So it falls off without replicating the last 100 nucleotides or so at the end. Let's see, common abbreviation here, base pairs. We call them base pairs because normally in the DNA double helix, they're paired up with two strands. So every time a cell divides, the telomere on the new strand becomes shorter by about 100 base pairs. And it takes about, for human cells, it takes about 50 cell divisions before the telomere is completely gone. And when a normal cell, has no more telomere left, it will stop dividing. And that's the Hayflick limit. Not every cell obeys the Hayflick limit, though. So some cancer cells will continue to divide even when the telomere is completely gone, so they ignore the signal to stop dividing when the telomere is gone. Other cancer cells produce an enzyme, telomerase, that is another DNA polymerase that specifically makes the telomere longer. So certain cells have telomerase in them normally, so that means every cell in your body has the gene for telomerase. It's just normally turned off, except in germline cells, like cells that are going to make eggs and sperm, and a few other stem cells can lengthen their telomeres. So in germline cells, telomerase, every time the cell divides, telomerase, which remember is another DNA polymerase, comes in and re-lengthens the telomere back to its full length. So some cancer cells have mutations that turn on the telomerase gene, which should be off in your normal body cells. So we can add telomerase to culture cells. This is how it was discovered that the Hayflick limit was due to telomerase, is that you can add it to culture cells. So down here, this little blue line, that's what normally happens. That's the Hayflick limit. So we have a number of divisions here. And normally, human cells, even if you get them from an extremely young individual or a newborn or even a fetus, they'll only divide about 50 times and then they stop. If you add telomerase, you can get human cells to divide indefinitely, like cancer cells. But there's more to cancer than just over-replicating. Cancer cells lose the characteristics of their original tissue type. They no longer function as part of the tissue in which they are found. They interfere with the surrounding tissue and don't allow it to function correctly. But that's one of the things they're looking for when they take a biopsy. They're looking at the cells specifically. Have they lost the characteristics of their original cell type? Are they invading the neighboring tissue? This picture down here, this is a picture of a lung cancer tumor that's at a very early stage. And you can see the little sacs around it of the normal lung tissue, and then this little blob of very small cells which have clearly lost their tissue type. They are no longer looking like the normal lung cells that are around it. We refer to a tumor as malignant, so we can see the Latin root in here for bad. Mal means bad. So malignant tumors, that means they're invasive. That would be a cancerous tumor. A malignant tumor is a cancerous tumor. And that means it's invading the surrounding tissue. It may metastasize, so that means that cells leave the original tumor and seed new tumors in other parts of the body. And those are referred to by the noun form of that would be metastases. If there are metastases in the body, that means that you have tumors that are coming from, that are not the original tumor. They've been seeded there from the original tumor. So that's what metastasize means. It doesn't have to metastasize for it to be considered cancerous, though. For it to be considered cancerous, it has to be invasive. It has to be invading the surrounding tissues. So we have proteins that try to prevent cells with mutations from getting out of control. We have DNA repair mechanisms. So DNA gets damaged all the time. DNA can be damaged by radiation, by chemicals. There's lots of things that will damage DNA. Errors just occur during DNA replication. And there are DNA repair proteins that are constantly, in the nucleus anyway, that are constantly trying to fix the DNA in the nucleus. And one of those proteins is P53. P53 is known as a proto-oncogene because it's commonly mutated in cancerous cells. But I think it's more useful to know what this protein actually does, because when you hear a gene is an oncogene or a proto-oncogene, onco means cancer, it implies that this gene is somehow bad. But in fact, these genes have really important functions in the cell, and P53 is one of those. So you only have 2 copies of P53 in your cells, one on each of a pair of homologous chromosomes. And if both copies of the P53 gene are damaged because of mutations, because it's cancer, then you're missing this important function. So what P53 normally does, it 2 things. One thing is it triggers repair of DNA. So if there's a break in the DNA from radiation or chemicals or whatever, it will trigger repair. If the DNA is not repaired, then it triggers the cell to destroy itself through a process we call apoptosis, which the cell breaks up into little vesicles that are then consumed by white blood cells. So that's what should happen. Now, if you're missing P53 or you have mutations in both of your P53 genes so that the protein that's produced is abnormal and doesn't work, now we have cells that are accumulating mutations, they have DNA damage, and now they can just continue to divide. So missing P53 itself doesn't cause a cell to become cancerous, but what it does do is allow those cells to survive. Cells that are accumulating massive amounts of DNA damage will just continue merrily along dividing, and then it's possible that at some point in the future that cell may become cancerous. It will continue to accumulate mutations and might eventually become a cancerous cell. Another way to look at cancer, instead of focusing on all of the different mutations and the different tissue types of origin, is to ask, what do cancer cells have in common? And this is becoming a more of a question of interest in the last 10 years. A long time ago, a scientist named Otto Warburg discovered in the 1950s that almost all cancer cells, in fact, do have something in common, and that is a deranged metabolism. They don't use oxidative respiration as their primary mode of getting ATP. Instead, most of their ATP is coming from fermentation. So if you remember from respiration, oxidative respiration takes place in the mitochondria. This down here on the lower left, that's a healthy looking cellular mitochondria. And most of the ATP in a normal, healthy human cell is coming from oxidative respiration in the mitochondria. But during intense activity, like if you're doing wind sprints or you're lifting heavy weights, your muscles are going to be fermenting. And that means that they're going to be getting most of their energy from glycolysis, which takes place out in the cytoplasm. So cancer cells have been found to contain defective mitochondria most of the time. And this on the right here, that's an example of a defective looking mitochondria. Sometimes they're called ghost mitochondria because they're missing a lot of those folded membrane, the inner membrane that we see in the healthy mitochondria. This effect, the fact that cancer cells that are growing in culture are mostly using glycolysis and fermentation, that's called the Warburg effect now. So people have asked, well, does that mean anything for treatment? It certainly means something for detection and has been used and still is used to detect cancer cells. So you might know that cancer cells are very glucose hungry, and if they want to find out if you have, once a cancer has been detected, if they want to find out if you have any metastases anywhere else in your body, they'll typically do a PET scan, PET scan. And what they do is they give you some radioactive glucose to drink, and then they wait about a half an hour, and then they see where the radioactive glucose is in your body, because cancer cells are going to suck up that radioactive glucose, and that radioactivity is going to be found near the highest metabolic activity cells, which would be cancer cells. And so because they are fermenting, besides sucking up glucose and needing a lot of glucose, they also end up with a lot of lactic acid or lactate, which you might remember is the byproduct of fermentation in human cells. So what they do is they dump all that lactate out of their cells, because the way a normal cell is going to get rid of lactate, well, there's a couple of different ways, but in muscle cells, if you do a cool down after you do some intense anaerobic activity, the mitochondria in your muscle cells are actually going to suck up that lactate and burn it and fully process it to carbon dioxide. But it can also go into your bloodstream, can end up being processed by the liver, and that's what happens with cancer cells. They dump all that lactate into the fluid around them, and they rely on healthy cells around them to get rid of the lactate and for it also to go to the bloodstream. So there are some clinical trials going on right now for brain cancer, and there's one that's going to start soon for pancreatic cancer, looking at if starving cancer cells of glucose using a very carbohydrate restricted diet, an extremely strict type of ketogenic diet, in addition to standard of care with chemotherapy and radiation, is effective. And it's looking like that's the case, that ketogenic diet alone is not looking like an effective cancer treatment. However, using that in addition to the radiation and chemotherapy looks like it makes those treatments much more effective. So expect some new information along those lines to come out. Clinical trials are going on. Expect new information on that to come out in the next year or two. So if we compare a cancer cell to a normal cell, cancer cells are growing out of control. They don't mature into whatever tissue type they're supposed to be. They have evaded the immune system. Normally, we know the immune system is involved with early cancer surveillance and preventing cancer. But once there's a tumor growing, that means your immune system has not recognized it as being not self. Tumor cells don't require oxygen, which is one of the ways they get around not really needing an excellent blood supply, because they don't need to have a continuous supply of oxygen, because remember, they're not using the mitochondria for that. They still use their mitochondria, but the Krebs cycle, the citric acid cycle in the mitochondria, is instead being used to create metabolic intermediates to build amino acids, fatty acids for phospholipids, etc. They're still using them, but they're not using it to generate ATP. They're using it to generate metabolic intermediates that can be used to construct cellular components, because that's the two things that cancer cells are doing. They do need energy, and they also need building blocks to make new amino acids to build new proteins. They need intermediates from the Krebs cycle to build fatty acids and phospholipids. So cancer cells tend to create an acidic environment around them thanks to the lactate that they're constantly oozing out of them because they're not using that, and their nutrient preference is glucose. Other cells don't have to rely on glucose. The other cells in our body can use fat and ketones as a source of energy as well. And if the cells are using oxidative metabolism, which would be all of our nervous tissue all the time is doing that, and our other tissue is doing that almost all the time, unless you're doing some kind of anaerobic activity. They require oxygen, whereas cancer cells do not. So some of those things that we talked about that control the cell cycle, these have been targets for cancer treatments. One of the most effective targets of cancer treatments has been stopping this initial signal here with a growth hormone, and the most effective ones have been when that signal is estrogen or testosterone. Now, those receptors are not on the cell surface because those are fat-soluble steroid hormones that go all the way into the nucleus. But that's been an effective treatment. So if you have a breast cancer or a prostate cancer or a uterine cancer, one of the things they will do is test to see if your cancer needs those hormones, needs estrogen or testosterone in order to grow. If it does, then that means that can be an effective treatment. The other types of cancer treatment just focus on preventing cell division, either by preventing assembly of microtubules or other things, and they just stop cell division just globally in the body. And that's one of the reasons that chemotherapies often make people's hair fall out and cause digestive upset, is because hair follicles and the lining of your intestines and your stomach and your esophagus, those are rapidly dividing tissues in the body. So if we give a drug that is globally slowing down cell division, it's going to affect all of those. It also tends to make people anemic because of the same reason, that there are rapidly dividing cells in your bone marrow that are resupplying your blood cells, and those are also going to be affected by that. Contagious cancers are known, and most of those are caused by some kind of a virus, and there's quite a few of them known, especially in birds. There's a lot of contagious cancers that are known that are caused by viruses. The one that you certainly have heard of is the human papillomavirus, or HPV, which causes warts. There are dozens and dozens, I think they're close to 100 now, different varieties of human papillomavirus that cause all different kinds of warts. Almost all of them are benign. But there's a handful of HPV viruses that cause cancerous growths. And these are mostly cervical cancer in women and throat cancers in men and women, and then a smaller number of penile cancers in men. And we now have a vaccine which is continuously being updated as new varieties of the virus are identified. The vaccine, which is one of the brand names, is the Gardasil vaccine. If you're under 30, I think anybody under 30, male or female, can get the vaccine. Initially, it was recommended only for girls and women because cervical cancer was the primary concern, but now it's been found that of the throat cancer victims of HPV, most of those are men. So they're now recommending the vaccine for both men and women. For women, the greater risk is cervical cancer. For men, the greater risk is throat cancer from HPV. And so if you're under 30, you can get that vaccine. It's a series of 2 shots, and it's extremely effective at preventing cancer from one of these varieties of the virus. Now, if you have a wart, you should not be concerned because Most of HPV don't cause cancer. They just cause warts, or they cause nothing, not even a wart. But if you're concerned, if you're under 30, you can get the vaccine. Another virus-caused human cancer, which was only discovered because of the AIDS epidemic, is that Kaposi sarcoma is caused by a herpes virus. Before the AIDS epidemic, Kaposi sarcoma was thought to, it's a kind of a skin cancer where you get like purplish patches on your skin, which eventually start to die. Kaposi sarcoma was thought to be a genetic cancer that it was found mostly in elderly Greek men, and it was thought to be genetic. And then AIDS patients started getting it. And then it was discovered that it's caused by a herpes virus 8, which normally causes nothing in anybody. You have to be somewhat immune compromised for herpes virus 8 to cause Kaposi sarcoma. So that's why it was seen in elderly populations, and then it appeared in AIDS patients because they're severely immune compromised. Another type of contagious cancer is not caused by a virus. Instead, cancer cells move from individual to individual. And now this is only possible in populations where there's very low genetic diversity, where individuals are inbred and related to each other. There is a cancer like this that's in dogs, and also Tasmanian devils. This is a Tasmanian devil. They are a marsupial mammal. They are one of the only remaining carnivores in the marsupials that are left. Tasmania is an island off of Australia, and that's where they were originally found. And it was discovered about 20 or 30 years ago that they had this apparently contagious facial cancer, where they would get these large tumors that would eventually prevent the animal from eating or drinking, and then they would die. And it seemed to be contagious, so initially they looked for a virus, but it turns out that it's the cancer cells themselves that are being transmitted from individual to individual because of a peculiarity of Tasmanian devils. They're pretty ferocious, and when two devils meet, they will often get into some sort of a fight and bite each other on the face, and that's how the cells were being transmitted. These tumors tend to be kind of fragile and slough off cells quite easily. So if an animal with a tumor bites another animal on the face, some cells would slough off of the tumor into the open wound, and then they would start to grow. And now this is only possible because Tasmanian devils were decimated by hunting, trapping, and poisoning, and the remaining the population that's left is very closely related. So that's the only way this could occur. I'm not aware of a human cancer like this that would be transmitted as cells from individual to individual. Cancer cells are extremely useful to science because they don't have the Hayflick limit. So we can grow them indefinitely in culture. We can use them to study cell division. We can use them to study different types, tissue types, because there are cancer cell lines that are available for purchase that are from all different tissue types. And although they're not the same as normal, if you want to study something in liver, you can look at a liver cancer cell line that will be easy to maintain indefinitely in culture. And so we call these immortal cell lines because they're not going to obey the Hayflick limit, and they're just going to keep dividing. The HeLa cells are the most common immortal cell line. They were the first immortal cell line. They are extremely useful as well if you need to study a virus, because viruses have to reproduce in a live cell. And if it's a human-only virus like polio, you have to have human cells to grow it on. And HeLa cells were very useful for that. We now know that the HeLa cells came from a cervical cancer in a woman, Henrietta Lacks, who there's a book and a movie about the development of this cell line. and the family of the woman whose cells were used. And we now know that her cervical cancer was in fact caused by the human papillomavirus. The DNA of that virus is found in the genome of HeLa cells today. But it's a very interesting story. The book is called The Immortal Life of Henrietta Lacks, and there's also a movie, not a documentary, a movie by the same name. So again, so for this chapter, you should know what happens at each stage of the cell cycle, G1, S, and G2 of interphase, what is happening in prophase and prometaphase, and we will consider prophase and prometaphase to be one process for the exam, because we're not going to distinguish too much between what's happening in prophase and not prometaphase, because it's really just because prophase is really long that we divide it up into two, early and late. So nuclear membrane breaks down, chromosomes condense. The replication of the centrioles actually occurs in G2. Replication of the DNA occurs during S. So those important processes, which are important parts of mitosis that occur during interphase, you should know those as well. By the end of prophase, we have the spindle formed, the nuclear membrane is completely broken down, and all the chromosomes are now attached to microtubules. In order for it to be considered metaphase, we have to have the centromeres all on the metaphase plate. All of the sister chromatids separate and move, and that's anaphase. And then telophase is describing what is happening to the nucleus. So the nuclear membrane is reforming during telophase. Cytokinesis is what's happening to the rest of the cell. So cytokinesis is the cytoplasm and the cell membranes separating. They are not the same, and they don't necessarily occur at the same time. We can have telophase without cytokinesis, which we had some examples of that.